I want to talk to you for just a second about some molecular studies, so hang in there with me. These are called linkage studies. And linkage studies are studies that can be done to try to match the genes in different groups. So here is an X chromosome. The X chromosome has over 1,000 genes. The X chromosome looks like a little skein of yarn here, and that's a good analogy because it is a wound up chain of DNA. So if you could unwind this, you would find almost 1,100 genes on this one chromosome. So the method that's used to find these matching regions is called linkage studies. So we're looking for the same trait on different chromosomes. So what this is that there are probes that you use, and you're trying to match regions in gay brothers that are not there in straight brothers. So if you can have those two groups and you can separate them very well, then you do these linkage studies in order to find those traits that are characteristic of the gay brothers that are not characteristic of the straight brothers. There is a diagram down here as well, and this just shows for the X chromosome all the genes that have already been found on that and described, and there are different segments here. Each one of these segments has a different number, and so the segments are from 1 to 28. The values that you need to be looking for are anything over 50%. If it's over 50%, this is indicative of linkage. It's saying that there are genes there in common. So let me give you a rundown on one of the best studies that has been done in this regard. The study population here consisted of 33 families with 2 non-twin gay brothers and 36 families with 2 non-twin lesbian sisters. Genotyping was done. Genotyping is the probes for these different areas of the chromosome, and there were 8 different areas that were looked at in 3 regions of this distal long arm of the X chromosome. What happened? When gay brothers were studied, there was 67% linkage. When gay and straight brothers were compared, there was 22% linkage. And I'm telling you, anything over 50% is significant. The difference between 22 and 67% is very highly statistically significant. However, look at the data with lesbian sisters and lesbian and straight sisters. They're both just a little over 50%. They are not different. And so you have to conclude that while you see a genetic linkage in gay males, you do not see a genetic linkage in gay females. The greatest amount of linkage was in that very last part of the X chromosome, an area called XQ28. And this, again, has been confirmed by even 2 more recent independent studies done in different laboratories with different investigators. Very interesting situation in which there seems to be more genetic component with gay males than with gay females. So what are the conclusions to date from these genetic studies? Well, we've found linkage in that XQ28 region, and if you take all the studies together, there have been over 22 markers that have been used. That makes them very strong studies. If you look where that little circle is, those are the X chromosomes, and it's that end, that X Q28 region that has been implicated. However, if you look at animal studies, in this case from mice, you'll see that there are many, many genes that have been implicated. 51 genes that are involved in sexual development, and 7 of those genes show the greatest effect on sexual orientation and behavior. Not only did they find them on the X chromosomes, but they're found on chromosomes 7 and chromosomes 10. It is very likely, I would place money on it, that there will be many different genes that will be implicated in human sexual orientation, and they are unlikely to be located on one chromosome. They're more likely to be found on many chromosomes, just like we're finding in the animal studies, because it's a very complex situation. And anytime you have a complex biological characteristic, you can almost be assured that you're going to have many genes involved.